R11s and 12s. This is going to be the second video in the nucleic acids PowerPoint when we're focusing on transcription. This is for the IP diploma, remembering this is the HL content for nucleic acids. This time, like I said, we're going to be looking at modifying messenger RNA and all the different post-transcription modifications that happen in order to create a protein in translation. And we'll talk about translation next week, but this week we're looking at transcription and modifying mRNA. Let's begin. So, we talked about the fact that eukaryotic cells will modify mRNA after transcription. That's one of your understandings, which is here in the bold in yellow. There's actually 3 post-transcriptional events that we need to look at and memorize for your exam coming up. That's capping, polyadenylation, and splicing. Capping is when we add a methyl group to the 5' prime of the RNA strand. So, as you can see here, it's the 5' prime down here, and we add this cap here. Okay, so we're adding a methyl group, which you can see here, methyl group. Okay, and the methylated cap, the whole purpose of that is it provides protection against degradation. So, an enzyme called exonuclease can't come in and remove the successive nucleotides from the end of that molecule. So it prevents the basically the RNA strand from being broken down by exonucleases. It's also useful, we don't know in detail, but it's also useful for the ribosome to recognize the transcribed strand. Okay. Polyadenylation, on the other hand, is when a long chain of adenine nucleotides, which we can see here with the repeated A's, is added to the 3' prime of the RNA strand. It helps to increase the stability of the RNA transcript, and it also helps the process of physically leaving the nucleus, making the way to the ribosome. Okay, so on the 5' primed, we've added our methyl group to prevent degradation. On the 3' primed, we've added our long chain of adenines in order to help it leave the nucleus. The main one we're going to talk about, though, is splicing. So, mRNA splicing, we've talked about before. A gene physically contains something called introns and exons. Now, the exons are the coding part of the gene, so they're helping to make the protein, and the introns are the non-coding parts of the genes, non-coding for the protein. They still have a point, and we'll talk about that later. Okay. Splicing's job is to remove those introns. Since they're not coding for a protein, they're not needed at the ribosome, so splicing's job is to remove those introns from the RNA strand. Okay, and we can see it in this little picture here. So everything that's a light blue is an intron, everything that's dark blue is an exon. So once splicing takes place, we're physically removing the introns and leaving only the exons between the capping and the polyadenylation on the 5' and the 3' end of the messenger RNA. But that's not to say that splicing can't physically remove exons. So yes, its job is to remove introns, but it also helps us go through and increase the different amount of, or the number of different proteins that an organism can produce, which again is another one of our understandings. So that's when we're talking about alternative splicing. So that's when splicing physically removes an exon to code for a different protein. And we can see this here. Again, we've got 5 different exons here. We've got the green, blue, purple, orange, and red. Okay. To make protein A, we need all 5 exons. So no exons are removed in this splicing here. But to make protein B, we don't need exon 3. So the purple one, we don't need exon 3. So as well as all the introns, which aren't highlighted being removed, the spliceosome, which is the protein that helps with splicing, has also removed exon number 3, leaving us only with 1, 2, 4, and 5. That leads to protein B. So we've used the same gene, but created a second protein through alternative splicing. And we can see the same example here with protein C, where this time we haven't used exon 4, so the spliceosome has removed the introns as well as exon 4, leaving just 1, 2, 3, and 5 to create protein C.